Okay, let's begin our session 3, Research to Support Guidance Development for Inhalation Drug Products. Our first speaker will be Dr. Liang Feng Hang, who is a clinical analyst from the Office of Research and Standards within the Office of Generic Drugs. Then we'll have Dr. Ross Wallinger, who is our senior chemical engineer at the Office of Research and Standards within the Office of Generic Drugs. And then the third speaker we will have is Dr. Changnian Guo, who is a supervisory chemist at the Office of Pharmaceutical Quality Research, OPQR, within the Office of Pharmaceutical Quality. So let us welcome our first speaker, Dr. Liang Feng Hang. Thank you.
Hi, good afternoon. I'm Liang Feng Han. I'm the clinical analyst at the Division of Therapeutic Performance 1, Office of Research and Standards, Office of Generic Drugs. I will start this session by discussing FDA's general considerations using the alternative B approach in lieu of comparative clinical endpoint B study for suspension-based metered dose inhalers. This presentation has 3 objectives. First, I will recognize the challenges with conducting the clinical B studies for orally inhaled drug products. Then I will discuss the available tool and the data from FDA-funded research and the external inputs that contributes to our development of alternative B approach. Finally, I will list some recent PSGs for suspension-based MDIs that recommends alternative B approach and discusses their study design considerations. For local acting MDIs and DPIs, FDA traditionally recommends an aggregated weight of evidence to establish B that includes a clinical B study. But we know there are many challenges for the clinical B study. For example, these studies has high variability and the lower sensitivity due to the flat exposure response. As a result, these studies usually requires large samples and a long study duration. Consider the huge cost and the long time required for conducting these studies. Now FDA considers an alternative B approach in lieu of the clinical B study that includes in vitro, in vivo, and in silico methods. Ideally, the alternative B approach could assess each step of the local drug delivery from pre-actuation to the local deposition. These steps include the formulation and the device characteristics before actuation. And after actuation, the aerosol is formed. So after the aerosol leaves actuate, the droplet size changed due to the propellant or other excipient evaporation. After the aerosol enters airway, how the patient's anatomy and the physiology condition affect the local drug deposition. So after the final drug local deposition, the dissolution and the absorption from the local site. And we now have many in vitro studies, such as realistic APSD, dissolution, and MDIS that can provide the information for each step during the process. But we have limited in vivo study methods and some in silico study methods that provide information on some of these steps. And the alternative B approach was first introduced in the PSG for the solution-based MDIs, like Cuba and the Cuba Radiator. The PSG states that if the test formulation is Q1, Q2, same as the LD formulation, and the test device is similar to the LD device. And the applicants can take an alternative B approach that includes characterization of emitted sprays, morphology imaging comparison, and the real-risk APSD dissolution study, and the in silico modeling, and the alternative PKB studies. And after that, the next question is that can we use the same approach for the suspension-based MDIs and DPIs? To answer this question, FDA hosted a 2-day workshop that invited experts from industry and academics. The workshop consisted of the larger conference and the small group discussions. At the workshop, FDA presented the thinking on the alternative B approach and received feedback from the external experts. The consensus at the workshop was, in general, the alternative B approach for the solution-based MDI could also be used for the suspension-based MDIs and the DPIs. But some studies, like the real-risk APSD dissolution in silico methods, are more useful to establish B than other studies. And the particle morphology charcoal block PKB studies are partially useful or for the confirmatory purpose. Evaporation rate and the velocity profile evaluation, pre-actuation characterization of the formulation have limited utility. It should be noted that the alternative B approach is product specific. That means not a single alternative B approach that works for all the products. So after the workshop, the FDA compiled the outcomes from the event and posted the new PSGs for the suspension-based MDI. All these PSGs take the option-based approach. If the test product, a test formulation is Q1, Q2, the same as the LD formulation, and the test device is similar to the LD device. Oh, shoot. That the option 1 includes that the conventional in vitro B studies, the in vivo PKB studies, and the alternative B approach include additional in vitro B studies, characterization studies, and the charcoal PKB studies, and the in silico studies. If the test formulation is not the Q1, Q2 as the LD formulation, and the test device is similar to the LD device, and the end applicants can take option 2 that follows traditional B recommendations that include a clinical study. And the option-based approach was first introduced in the PSGs for generics referencing Bavaspi aerosphere and the Bradstreet aerosphere. There, FDA approved the suspension-based MDIs indicated for the COPD treatment. Their formulation contains this DSPC and the calcium chloride, and these components contributed to the formation of the phospholipid-based porous particles. FDA posted the 2 PSGs for Bavaspi and the Bradstreet aerosphere in this February, then posted 4 PSGs for other suspension-based MDIs this August. For option 1, the test formulation, as I mentioned before, should be the Q1, Q2 the same as the ILD formulation. The in vitro BE study included the conventional in vitro BE study and the 2 additional in vitro BE studies that are realistic APSD and the dissolution. Comparative characterization studies, such as particle morphology of the emitted dose, the in vivo PKB study, and with and without the charcoal block. And the in silico modeling is optional for the BE establishment, and the test device should be similar to the ILD device. As I mentioned before, the alternative BE approach is product specific. You can see here is the dissolution study is only recommended for three products with slow dissolving APIs. And the morphology study, particle morphology study is only recommended for 2 products that can have that with the porous structure. For option 2, the test formulation could be Q1, Q2, or not Q1, Q2 to the ILD formulation. The in vitro BE studies contains the conventionals at the in vitro BE studies, and the comparative characterization study including the particle morphology, and the in vivo PKB study without charcoal block, and include a clinical BE studies. And the in silico modeling is also optional for the establishment. The test device should be similar to the ILD device. And you can see here that the option 2 is the same for all the products. Okay, now I will discuss the study design considerations for each alternative BE study. The GDUFA funded research outcome indicated that there are many factors that affect the APSD study. For example, the mouse-throat model size, the inhalation profile, and there's other factors including the finding time point, and the angle of the mouse-throat model inserted. And we also noticed that the response to the various study factors is product specific. And in this figure shows the fine particle fraction variability. It's that for the different products, when different size of the mouse-throat models were used in the realistic APSD study. Therefore, we recommend that your method development should consider the mouse-throat types and size and inhalation profiles and other factors. This figure shows that the different mouse-throat models and the realistic inhalation profiles and the constant flow rate inhalation profiles. We recommend that the realistic APSD can be conducted at the beginning life stage. If the conventional APSD study shows that no difference between the beginning stage and the end stage. And the applicants also can take a bracketing approach. For example, the small and the larger size mouse-throat models plus weak and strong inhalation profiles. As long as the combination of the mouse-throat models and the inhalation profiles can cover the expected inter-subject variability of the indicated patient population. Alternatively, you can justify your choice by showing good in vivo correlation if you have this data. And the inhalation profiles should be obtained from patient. And we see some submissions that they provide inhalation profiles from the health subject, and we think it may not be appropriate. And the B is based on the PBE analysis of the impact size mass for each mouse-throat model and the inhalation profile combination. And the FDA welcomes that alternative statistical approach if this approach is can be scientifically justified. And you can also request a pre-end meeting request to discuss your proposed statistical approach. Post dissolution study, we also found that many factors can affect the dissolution study, especially the particle size of the emitted particles. And we noticed that currently there is no standard methods. And the method development could be product specific. And in this figure at the bottom, you can see that this figure shows a good dissolution study, because this dissolution study can distinguish the different dosage form and the different particle size. And we also acknowledge that we also acknowledge that the need for the dissolution study is product specific. For fast dissolving API products, the dissolution study may not be necessary. And the PSG recommends the dissolution study should be conducted at the beginning of the life stage. And the study should collect the aerosolized dose of similar drug mass between the T and the IS products. And your method should be optimized and validated by modulating your apparatus sample collection and the dose, etc. And your dissolution study must be discriminatory, that can identify the difference in the deposited drug particle size. And the B is based on the comparative analysis of dissolution profiles with inappropriate statistical methods such as F2 analysis. And after inhalation, some OIDPS could be swallowed into the GI contract. If there is a significant GI absorption from GI contract, the conventional PKB study without charcoal block cannot distinguish the systemic exposure from the inhalation portion or the swallow portion. Therefore, for these products, we recommend a PKB study with charcoal block that can eliminate the GI absorption and obtain the PK profile only from the local lung deposition. And we understand that the charcoal PKB study with or without charcoal block have many similarities in the study design, such as how subjects are recommended for the study, minimum number of inhalations, etc. And currently, the only difference actually is the administration of charcoal block. But currently, there is no standard for the charcoal dose and the timing of the charcoal administration. So that's therefore that your method for charcoal administration should be justified in your end submission. And the B is based on the 90% CI for the TR ratio for the AOC and the Cmax being between 80 to 125%. Now we think that the role of comparative characterization study provides supportive evidence for establishing B between the test and the RD products. For example, that the particle morphology can contribute to the meaning that the in vitro study, the drug performance, such as APSD and the dissolution performance. And in this figure, we see that we have 4 drug products with the same amount of the fluticasone. But the morphology, so the imaging study shows that there's different free fluticasone and the fructose associated with lactose. And these different association status can be detected by the dissolution study on the right. And we also, again, said the recommendation for the morphology imaging is product specific. Currently, just as I showed before, that currently this study is only recommended for the products with porous structure. The PS recommended 3 batches of T and IF products and tested at the beginning stage of the product. And the imaging in comparisons should be conducted on the deposited particles of the emitted dose. The morphological features of the particles, which may include the agglomeration and characteristics, should be evaluated. A description of sampling collection method should also be provided. So, and at the end of my talk, I prepared 2 questions for you. Question 1, which of the following statement is not true? So we have 3 options, and wait a little bit, and then review the answer. So the correct answer is C. All the studies in the alternative BE approach need to be conducted for BE establishment for most suspension-based MDIs. This statement is not true. Question 2, which of the following statement is true? We also have 3 options. And the correct answer is C. For suspension-based MDIs with limited GI absorptions, charcoal block PKB study may not be necessary. So in summary, the challenges with conducting clinical BE studies could lead to high cost and longer drug development. To address these questions, FDA has explored the in vitro, in vivo, and in silico study design through the GDUFA-funded research projects to identify the alternative approaches that can be used to include the clinical BE study. And following the FDA-sponsored workshop on alternative BE approach, FDA has utilized the input received from industry and academic attendees to aid the development of several PSGs for suspension-based MDIs. And this development, the PSGs, present FDA's efforts to expand alternative BE approaches beyond just the solution-based MDIs, and highlighted the additional study consideration needed to apply to the suspension-based MDIs and also DPIs. And so I will wrap up my talk and turn to the next speaker, Dr. Ross Olander. Thank you.
Hello, it's my privilege to be speaking to you today about the in silico recommendations in our recent PSGs that Dr. Han alluded to. So I have several learning objectives for today's talk. The first is that by the end of the talk, I'd like for you to be able to identify recent product-specific guidance or PSG documents that include detailed language regarding the use of in silico modeling for orally inhaled drug products or OIDPs. The second is I'd like for you to be able to list potential roles for in silico studies within alternative bioequivalence or BE approaches for OIDPs. And finally, I'd like for you to be able to describe recent PSG recommendations for in silico study design and model credibility establishment. So as Dr. Han mentioned, there are some new PSGs that were published this year in February, and they're listed here. He spoke mostly about MDIs, but we did have new recommendations for 3 DPIs as well. And on all 5 of these PSGs, there are recommendations for the use of in silico modeling. So the first of the two options is an option that does not include a comparative clinical endpoint or pharmacodynamic BE study. And the PSG for formoterol fumarate glycopyrrolate is the one I'd like to draw your attention to because this has the detailed language for computational modeling. All the others just sort of refer back to this PSG. So computational modeling is not included in the two BE options, but there is detailed language given there to clarify purposes for why you might want to use a model. So what are the two potential roles that we've identified in our PSG? The first is to identify biorelevant limits for BE comparison of key recommended studies by establishing correlations between results of in vitro and or in vivo studies and in silico regional deposition predictions, and also to conduct virtual BE trials using regional deposition predictions. And I'm going to get into that in a little bit more detail later on. So if you have an in silico study that you'd like to use for your application, I've got some details here about the design. So first, a little bit about the different types of modeling you might use. You might use computational fluid dynamics or CFD modeling. And what this does is it allows for prediction of fluid and particle transport such that you can predict deposition in the different regions of the lung. It allows for consideration of realistic geometries such as a realistic mouth-throat or realistic lung geometry, and can be validated with in vivo or in vitro data. So I've got a couple of animations here. The animation on the top is showing the results of a CFD simulation, and this would be the prediction of the fluid motion. And then the bottom simulation here is showing the particle motion within that flow field. And then when the particles deposit, that's when you quantify deposition in the different regions. Another type of modeling is semi-empirical regional deposition modeling. So this is essentially doing the same thing that the CFD model does. It just has a different mathematical formulation using algebraic equations and some empirical formulations. Branch-specific deposition probability is the ultimate goal of this model. And then the deposition values are summed across the branch levels to obtain regional deposition. And this is an example of what this might look like if you have the results from a study like this. One thing to mention is that this was originally developed for toxicology purposes, and some of the equations have been changed a little bit to fit for a pharmaceutical application. So how might you use regional deposition modeling to support OIDP, generic OIDP? So as I mentioned before, biorelevant BE limits. This would be testing the sensitivity of deposition in central and peripheral lung regions to differences in vitro metrics, such as APSD with realistic mouth-throat models or plume geometry. And the purpose of doing this would be to potentially widen the limits for in vitro comparison. If you have an in vitro study that may be failing by a little bit, you may believe that when you propagate this into the lung, that it may not constitute an actual difference at the site of action. So you could potentially use a model to widen those limits a little bit. Also, virtual BE trials. So this could be conducted almost like another study and could provide additional assurance of BE, especially if you're taking an alternative approach to what's already recommended now, but outside of one of the two recommended options. One thing I'd like to mention is that it's important when you're building a model to connect with in vitro data, and this is applicable for PBPK2. For a deposition model, the APSD results from realistic mouth-throat testing or semi-empirical can be useful for semi-empirical or CFD models as inputs, but they can also be used as validation data to compare the mouth-throat deposition with. If you have a PBPK model, dissolution data can be used as an input, but also could be used to assess the biorelevance of the different dissolution methods. And other relevant studies are plume geometry and spray velocity. So with these studies, these can provide model inputs as well. And lastly, it's important to ensure that your modeling assumptions and results are not in conflict with the collected in vitro data. So for example, if you have deposition data in one of the mouth-throat models here, you would want to make sure that your model is matching that mouth-throat model. So how can we consider variability within deposition predictions? One way would be to consider a population modeling approach. And this was an interesting paper I found from a few years ago, where they used something called functional respiratory imaging to predict regional deposition in 20 different patient-specific geometries. And this technique allows for using high-resolution computed tomography scan data to define patient-specific inhalation profiles in addition to the lung models. What you have are inhalation profiles and lungs that are paired together. And then they predicted deposition in 2 different MDIs and then compared them. So you could think of, if you're doing this to support your application, it would be almost like an in vivo study where you're selecting the proper number of subjects to power your study. Another approach could be use representative models. And we have an example here from a GDUFA-funded research project to the University of Florida. And they predicted lobar deposition using CFD models. And they had 10 different models, 2 for healthy subjects and 8 for asthmatic subjects. And they predicted using monodispersed particle injections. So these results here are showing deposition predictions in the 5 different lobes for the 4 different particle sizes. You can see the lobes are abbreviated as LUL, LLL, et cetera. And you have healthy female HF, healthy male HM, cluster 1, 2, 3, and 4. And each cluster was considered to be a subtype of asthma. And you can tell that depending on the particle size or the lobe, that the variability could be very little or it could be very high. So by using this approach, you could have a set of 10 models that you use to just capture the population as a whole. You're not thinking about powering your study like an in vivo study. So I'd like to say some things about PBPK modeling and how it can be used to support these products. This is a compartmental modeling approach. It allows for prediction of both local and systemic PK by simultaneously predicting dissolution, absorption through lung tissue, metabolism, and by integrating with the systemic model. You would validate this using in vivo PK data. This figure on the left shows an example of this. And in this case, there were 2 predicted curves, and each had a different deposition input. So you can see that deposition can affect your PK outcome. So why might we use PBPK modeling? So one way would be to understand the role of regional deposition, because while we pretty much accept that regional deposition is important for these products, it might not always be a perfect surrogate for regional absorption because you have mucociliary clearance that can move slowly dissolving or slowly permeating particles through different regions. So that you can then use this to understand the impact of dissolution and permeation, and also potentially to predict regional absorption. This also can be useful for understanding relationships between charcoal block PK data and regional drug delivery. There have been several research efforts, both within and without FDA, to understand if we can take a charcoal block PK study and say, take some of the metrics to then use it as kind of a surrogate for regional drug delivery. So you could use modeling to support that kind of approach. I'd like to say a few words about model credibility. So this is making sure that your model is doing what it's supposed to do. And in our recommendations, we have some language about the American Society of Mechanical Engineers Verification and Validation 40 standard. This was a standard developed for computational modeling of medical devices, and we've repurposed it for pharmaceutical products. So a few ideas here. The context of use is something you might have heard before. This describes what question the model is addressing and to what extent. And model risk is determined by something called decision consequence and model influence. So you can see this figure here. Decision consequence would be what's the consequence of a bad decision. So usually this is not really tied to the modeling itself. This is just like, say if we approved a bad generic, what would happen? So a high decision consequence would be for a product where it could result in death. That would be like the worst thing that could happen. Low risk would be just a minor inconvenience. But then there's also the model influence, which is how much are you using the model to actually make the decision. So if you're not using the model very much to make the decision, even if it is a high decision consequence, it might not be a high risk model because the model is only playing part of the role in that decision. And next, I'd like to say a few words about verification and validation. So model verification, this is ensuring that your numerical solutions to the relevant equations are accurate. So for most of these models, these are not analytical models. We're using some kind of complicated numerical approach to solve the equations. For example, for CFD models, this can involve comparing model predictions for simplified scenarios with analytical results. And one example is pressure drop through a pipe. So anyone who has engineering background probably is familiar with this example. Mesh and time step sensitivity, this is something sort of specific to CFD. But if you've ever seen a CFD model, this is something that's important. For PBPK models, this can look like checking the solver settings to make sure that truncation and round-off error have been minimized. So it's just essentially making sure that your model is working the way it's supposed to. But then for validation, for regional deposition, the question is, how do we validate these models and what data sources do we have? We've focused on in vivo data sources. So gamma scintigraphy is one source. This provides a two-dimensional image, and central to peripheral ratio can be quantified using this. There's also SPECT-CT and PET-CT, which can provide three-dimensional images. And this here shows an example of gamma scintigraphy where you can see the radio-labeled aerosol deposition locations. And on the bottom, there's different schemes for how you divide this into central and peripheral regions, which you can see can actually vary quite a bit from one study to another and can present another challenge. So one way we've thought about overcoming some of the limitations with comparing with in vivo studies is a lung mapping approach. And so like I'm saying, that these are two-dimensional schemes, but the problem is your lung is three-dimensional, right? So if you think about that C area in the middle, you actually have peripheral lung airways that are pointing out in that area. So you need to kind of differentiate where is the actual CMP regions. And this approach that we've found from a previous study was that you can take a CT scan and then overlay it on the 2D image and kind of figure out what percentage of each branch level is falling within the CMP regions. And we think that this provides a more precise way of comparing predictions with the in vivo data. And a little word on validation of CFD modeling. We think that this should be done in all the lung regions, especially central and peripheral. The lung region should be supported by in vivo data and clinical understanding. We do have some recommendations now that give some specific ideas about how to do this. And you might need a complementary model for the small airways because CFD typically is better suited to the larger airways. So it can be helpful to pair with a semi-empirical model. And I've got some results here from a previous study that show a good comparison with central and peripheral predictive values and experimental values. I do want to note that lung mapping was not used in this case. So in this case, it still showed a good comparison, but we still believe that it can be quite useful. So I'm coming to the end here. I got a few challenge questions for you. The first challenge question, what type of in vivo nuclear imaging technique cannot quantify lung deposition in three-dimensional space? Is it SPECT-CT, PET-CT, or gamma scintigraphy? And the answer is gamma scintigraphy is only two-dimensional. The other two are three-dimensional, although there are some limitations for understanding that. And we do have a research grant going on now to help better get information from those types of studies. Challenge question 2, which of the following is not a potential role for in silico studies within alternative BE approaches for OIDPs? First is conducting virtual BE trials for regional deposition predictions. Second is to predict potential adverse events following administration of the proposed generic OIDP. And third is to identify biorelevant limits for BE comparison of key recommended studies. And the answer is B, we're not predicting safety outcomes with these type of models. That's not the purpose. So in summary, there are 5 new PSGs that include language describing the use of in silico modeling to support development and approval of generic OIDPs. And of course, as Dr. Heim mentioned, there are another 4 that were revised last month. So we have 9 now in total showing these recommendations. And in silico models might be used to establish biorelevant BE limits for recommended in vitro and/or in vivo studies, or to conduct virtual BE trials. In silico study design should consider in vitro data sources and methods for capturing population variability. And finally, model credibility should be established using a risk-based approach, as MEV-NV40 is just a suggested method that you could use another method as well. And the call to action to you is to consider using in silico modeling to support development and approval of your generic OIDP. And I have many people to acknowledge and too many people to really call out, but just want to give you a sense for how many people have contributed to this. And final thought, just on the value of generic OIDPs. This was a quote from someone, and you can see the impact that it had on their life and their family just by the reduction in cost. And next, I'd like to welcome Dr. Guo to the stage.
Okay, thanks for the previous 2 speakers, gave very detailed information. That's a lot of information to learn and also laid a solid foundation for my talk. And here I will switch gear to give you an overview of the OPQR testing and research to support GSP development. First, here's a message from Office of Pharmaceutical Quality, OPQ. Everyone deserves confidence in their next dose of medicine. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. So through this, here's learning objectives of this talk. So through my presentation will inform you on FDA's own laboratory support on inhalation drug assessment. And you will understand the research efforts conducted at the FDA lab to support guidance development. And at the end of this presentation, I hope you can list several newly recommended in vitro studies as options to establish BE for inhalation products. First, okay, this topic is on OPQR lab support for GSP. So who are the OPQR labs? So OPQR stands for Office of Pharmaceutical Quality Research. It's a sub-office under OPQ, and we are a lab-based office having 6 divisions. The inhalation lab and team is under Division 2, so we are located in St. Louis, Missouri. In our inhalation lab, we have a full line of equipment to do testing and research for inhalation drug products, including delivery dose uniformity testing for MDI and DPIs, Anderson cascading impactor, ACI, next generation impactor, NGI, for aerodynamic particle size distribution, APSD testing. And we have non-impaction fast imaging method instrument SpreeView to measure spree pattern and plume geometry. And we have laser diffractions, and we have various type of microscopy, including Raman microscopy, 3D X-ray microscopy, SEM, AFM, and also some other FTIR microscopy. And we have dissolution capacities and to perform dissolution, diffusion, and permeation. So what do we do at OPQR labs? So at OPQR lab, first we are support regulatory actions. So we provide product testing and a method validation program to support NDA and ANDA reviews, and also support other type of regulatory actions. And we also conduct proactive research, first to evaluate emerging technologies for characterizing inhalation product performance. And we're also involved in develop novel methods to establish better in vivo in vitro correlations. And in the last few years, we put a lot of effort to conduct research to support a product specific guidance, PSGs, and especially focus on developing method for alternative BE approaches, including the 5 areas: elastic APSD, laser diffraction, particle morphology, and we're also exploring evaporation rate and the velocity profile for MDIs, and we'll also develop clinical relevant dissolution method for MDI and DPIs. So in this talk, I will give you some examples on the first 3 areas. First is on elastic APSD, which the previous 2 speakers already talked a lot. So I will focus on more details in for what you see in the lab. So traditionally, APSD was measured by cascade impactor. And the cascading impactor collect respirable aerosols with particle size less than 9 microns. So ACI is a stack of metal jets and plates to collect those particles. And between the cascading impactor and the drug product, there's a sample induction port, which shows in this picture is a metal piece with 90-degree straight bend. So it's nothing like the human anatomy. So the first area to improve to develop realistic muscle models, as the previous speaker mentioned, is to using bio-relevant muscle models, such as commercial available Alberta idealized throat, short as AIT, or models developed by Virginia Commonwealth University, VCU. And the VCU models, the 3D profile is available online. You can print it out using 3D printers using different materials. So the second area for the realistic model APSD testing is to utilize realistic breath simulation. And here the scheme is what we do in our lab to set the equipment up. Traditional way is just using a pump directly connected with a cascading impactor. So that only provides you a constant airflow. So it's not simulate the human inhalation profile. And by adding a mixing inlet and also utilize a breath simulation equipment and incorporate compressed air, we can set up simulated flow through the APSD testing. So by combining the bio-relevant muscle models and the realistic breath profile, a better RV-RVC are expected. Here is an example for our previous study. And this study was published in 2019 on AAPS Pharmacytech. So the study is to investigate solution MDI and the suspension MDI using different type of muscle models. So our result shows, first of all, the comparison between in vitro and in vivo result are product specific. And for the solution-based MDI, all the muscle models shows close match to the clinical data. Even the USP throat shows close to the clinical result. However, for the suspension MDI, the results are mixed. Overall, the bio-relevant muscle models here, VCU models, have a better chance to give you a better RV-RVC. So second area we spend quite a time on is to investigate the feasibility of a laser diffraction to be used as an alternative or orthogonal APSD method. Laser diffraction is a mature technique in a lot of industries. So when a particle passing through a laser beam, a larger particle will give a small diffraction angle, while a small particle will give you a larger diffraction angle. And the pros of this technique is it provides a rapid measurement. Normally, it delivers the result instantly. However, there's cons. First, laser diffraction measures volume-based PSD. Although it can be correlated to APSD, but still it's not a direct measurement. Second, is more important, laser diffraction is not able to differentiate API and excipient particles. So therefore, for complex drug formulation with multi-component, laser diffraction cannot be used as an alternative method for APSD. However, for some simple formulations such as the solution or dry powders with only API presented, laser diffraction have a very good chance to be an alternative APSD method. So there's 3 new products has been studied using laser diffraction. First is the Spriva Respimat. It is a soft mist inhaler. And the soft mist inhaler is a kind of product between MDI and nebulizer. The device is very similar to an MDI device. However, the formulation in the aqueous solution is similar to nebulizer. Since the formulation is a homogeneous solution, so their droplet size can be studied as a potential alternative for APSD. And our result, laser diffraction result, shows a clean measurement of the aerosol with a medium particle size around 3 to 5 microns from different distance, which demonstrate the laser diffraction could be have a very good chance to be an alternative method for APSD for this drug product. The second example is a breath-actuated nasal powder on Zitra Axial. For this product, it just have a special design, and the airflow is coming from the patient mouth to blow in the air into the device. And the airflow bring the powder out, introduce the powder into one nostril of the patient, and the airflow coming out from another. So the drug formulation here is around 15 milligram of API powder only. Since the aerosol are API particles only, so it also have a good candidate for laser diffraction to be an alternative APSD method. And our result demonstrated this point, and with a good clean measurement of laser diffraction, shows the medium particle size is around 30 micron. And the laser diffraction not always give you perfect result. And for the third example, is a breath-actuated, thermally generated inhalation powder. So for this product, we had some trouble. The main point is most of the drug product was released at the first half seconds of the aerosol procedure, and it's highly condensed. The concentration is so high, and the laser diffraction measurement was not valid at that concentration. So the detail of this study has been reported in the last year's SBIA workshop, and more details were published at the RDD 2024 proceeding. And the conclusion is the laser diffraction was not recommended as an alternative method, APSD method for this drug product, due to the limitation of the high concentration of API particles. So the third area I want to talk about is to using MDRS, morphologically directed Raman microscopy, to characterize particle morphology and API-specific particle size distribution. So to demonstrate Q3 sameness, the particle morphology and the API particle size distribution is critical. Here, when we're talking about particle morphology and the particle size distribution, we're talking about API, not excipient. However, the challenge are apparently for most of the product, API and excipient particles coexist in the formulation. And sometimes more than one API will be presented, and the API may have more than one polymorphic forms. And traditional techniques, such as laser diffraction we just mentioned, they cannot differentiate chemical identities of the particles. So Raman microscope is a good candidate for characterize particles, API-specified particle size and the particle morphology. However, normal Raman microscope is a very time-consuming process. And the MDRS provide a special strategy, specially designed for particle sizing characterization by implementing a morphology screening procedure before Raman confirmation. So the basic idea is that morphology filters will exclude as many as excipient particles as possible before time-consuming Raman measurement. Therefore reduce the measurement time by 10 folders. At our lab, our research is focused on development, optimization, and the validate of MDRS method on different type of drug formulations, such as nasal spray suspensions, dry powder inhalers, transdermal systems, ophthalmic ointment, and the cream. And we have 2 publications in 2019 and 2021 on nasal spray suspensions. And we also have a poster on MDRS application on creams in this SBRA. Hope you'll have time to visit our poster session. In summary, inhalation drug testing and research is an integral part of OPQR's work. And our OPQR lab-based research programs provide data to support guidance development, facilitate evaluation of generic drug applications, and allow for risk-based assessment of new drugs. Here, I want to acknowledge our past and current inhalation team members and our collaborators from OPQ, OPQA, and OGD, ORS, and OGD OB. Finally, challenge questions. First, which city is the OPQR inhalation lab located? A, Rockville, Maryland. B, Silver Spring, Maryland. C, Washington, D.C. D, St. Louis, Missouri. The answer is D, St. Louis, Missouri. Okay, that's an easy one. Second one is not difficult either. So, which of the following technique method is not considered as an alternative B approach for assessment of inhalation drugs? A, realistic APSD. B, solid state NMR. C, clinical relevant dissolution. D, particle morphology. The answer is B, solid state NMR is not an alternative B approach for inhalation. Okay, with that, I'm wrap up the presentation session. I will welcome the 2 speakers and the 2 other members to come here to host a Q&A panel session. Thank you.
Okay, we have our panel all set up. Thank you. And we welcome aboard Dr. Elizabeth Bilski, Senior Pharmacologist from Office of Research and Standards within UGD, and also Dr. Jen Zhu, Senior Staff Fellow with the Office of Bioequivalence from OGD. So we are running a few minutes late, so let's see how many questions you can get in, and also allow you a break to go do the graphic survey. Okay, any questions in-house, please? Online.
We have an online question for Dr. Liang Han. Does the agency suggest that it is acceptable for MDI to be non-Q1, Q2?
Yes, so as I mentioned in the talk, if it's not the Q1, Q2, we recommend that the applicants take the option 2 that follows the traditional BE approach, including the clinical study. But we also understand that it's possible that if it's not Q1, Q2 the same, the applicants can also take the option 1, the alternate B approach. The rationale for us is that to take the request that the Q1, Q2 the same in the option 1 is that we think is that the suspension-based MDIs are complex. It's likely that the non-Q1, Q2 formulation may cause the not the BE between the in vitro BE testing and the performance. So we recommended the Q1, Q2 the same in option one that reduce the likely of this possibility. But we definitely understand that it's possible that you develop non-Q1, Q2 formulation and you still see the BE between the test and the early products. But in that case, we suggest you submit the pre-end meeting request to talk with us and justify your formulation. And that's my answer.
Thank you. Next question, please.
Another question for you, Dr. Han. Are there any concerns related to standardization of the charcoal methodology irrespective of the drug product?
Yes, not a concern. That we know that currently there is no standard charcoal block that method established. And we think that you can develop your own charcoal block the method, as long as that your charcoal block method can adequately block the GI absorption. And the dose of the charcoal block is not too high that can raise the safety concern. As long as that the safety and the efficacy of GI absorption blocking is satisfied, we think that the possible that the charcoal block method could be justified.
Thank you. We do have a question in-house, please.
Marie Ducharme, Learning and Confirm at Université de Montréal. Just a comment about the charcoal block. I've seen many charcoal block studies where the charcoal block will deactivate the drugs going in the lung because the charcoal block is given too close to the inhalation dose and people do not, swish and swallow enough to remove the charcoal from the oropharyngeal area. So those studies are very tricky. But you can see it right away from the PK curve because, in the PK curve, normally for those type of drugs, you see clearly two peaks. You see the peak from the lung, you see the peak from the GI tract. And when the charcoal block is not well given, the first peak is decreased. Okay?
Yes, we also see that problem that just like you say, if the charcoal block is not properly administrated, it also blocks the lung inhalation. So there are some methods that can optimize. For example, that after you take the charcoal block, before the inhalation products was given, you can rinse your mouth, right? Swallow it, rinse your mouth, and that can reduce the lung absorption. Also, that you may consider use the maybe reduced amount, somehow reduce the amount of charcoal block, but I don't know if it's a timing, yes. 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 So the method, I think, definitely can be optimized. Thank you.
Thank you. Online questions, please.
We have a question for Dr. Roswalenga. What options are available to applicants to discuss issues related to in silico modeling for orally inhaled drug products?
Yeah, of course, there's the pre-end meeting pathway, which has been talked about at length in these workshops, but there's also the new MIE Model Integrated Evidence pilot program. So that offers a new way to talk about your model because you can also cover multiple drug products in those meetings. So you could have a model that you're trying to use across your whole suite of products, and we can discuss it in a more kind of holistic manner.
Thank you. And we do have a webinar on MIE on our SBIA Learn repository. Okay, next online question, please.
All right, another question for you, Dr. Arlinga. How can applicants develop regional lung deposition models when there are no in vivo imaging data available for the drug product of interest?
Yeah, thank you for the question. This can be an issue with some products. I've talked to some length about in vivo data, and there are some products out there where you've got literature results, but in some cases you don't. So I think if you've got that situation, of course, you could try to do the study yourself, but if that's not available to you, you could try a more what we call a platform modeling approach, where you have several similar products that you do have data for, show that you have credible models for those products. And then as long as the product you're considering isn't too much different, it could still be used as a supportive model.
Thank you. Next online question, please.
This question is for Dr. Bielski. Does the agency consider alternative BE approaches for dry powder inhalers if the proposed generic product is Q1 or Q2, the same as the RLD with similar inhaler designs?
So you're asking if alternative approaches are available for DPI products that have similar formulations. We always consider alternative approaches for any of our guidances. And if you pay attention to we've been going through a lot of revisions for inhalation products. So if you've been paying attention in this arena, you'll see a lot of change going on. So I highly recommend you keep checking those periodically. And of course, if you have a DPI formulation that's Q1, Q2 or non-Q1, Q2, you can always come to alternative approaches. And you may see some of these option-based approaches that you see for the MDIs also being proposed for DPIs as well. I think those are, of course, always things that we will consider having meetings with you to discuss those options. And, you know, as anything, what we're going to include for those options are all going to be product specific. So it's a really good way to come talk to us, especially if you have something you think you'd like to propose to us. We're more than happy to provide our scientific input on your proposal. Thanks.
Thank you. Next question, please.
Next online question for Dr. Zhenzhu. Are there any differences between solution and suspension MDIs in the BE recommendations when using an alternative approach?
Thank you for the question. So for the recommendation for BE testing, I think for the solution and suspension product, they include the in vitro testing and PKB testing and CCEP or alternative study. They are similar, but from the drug product perspective, the suspension product, because it's a colored system, it's you can say it's a thermodynamically not as stable as the solution product, which is dispersed at the molecular level. So that could give you some more challenges or more studies that need to be done to do the B study. Thank you.
Thank you. Next online question, please.
Another question for Dr. Han. Will the agency accept a study where certain in vitro tests may not meet BE requirements, but the PK study shows bioequivalence?
Yes, it's possible that in the submission, we sometimes see that the applicants did not pass the solution studies, but it passed the PKB studies. And in some cases, that we will wait consider all the evidence, and sometimes we think that the PKB studies shows that the in vivo dissolution and adsorption, and in that case sometimes, but it really depends on case by case, and we accept that the submissions that fail some in vitro tests. Yeah, thanks.
I'd like just to add to that, as well as Ross pointed out, if you're failing some of your in vitro BE tests, it's a really good chance to use modeling as an option to kind of prove that it's not affecting the regional deposition or absorption. So again, even if you think you're failing and you have questions about that, please come talk to us. We definitely like to hear from you where your main challenges are coming from and which in vitro tests are really causing you the issues.
Thank you. Do we have more online questions?
Yes, we have a follow-up to the question regarding charcoal. Would you recommend after taking the charcoal, the subject should take some water to rinse the mouth out or not? And if water isn't used, will the charcoal be diluted and have lost of the blockage?
Yes, actually, I think it's the good practice that you use water to rinse your mouth and swallow it before the drug administration. Of course, that you don't have to drink a lot of water to dilute the charcoal block efficiency. That is so. That means that you probably need to optimize your charcoal block method.
Okay, thank you. Next online question, please.
We have another question that came in addressed to Dr. Bielski. If Company A conducts an alternative BE approaches as per agreement with the agency, whilst Company B conducts a traditional BE studies, including a clinical endpoint study on the same generic product, will the agency consider Company B's ANDA more approvable?
Thanks for the question. I think there are multiple ways to show evidence of your local bioavailability. So I wouldn't consider one option better than the other option. I think if we have more than one option available, you can use the ones that are available. Even if we don't have those options and you have an alternative, we will also accept that. Again, there are some risks to take, right? As a company, if you're proposing something new, that might take a little bit more effort and more risk on your part because you're not sure exactly how the agency will take that. But if you discuss with us early on and you give the agency time to consider that, the more likelihood we may consider that early on. So I think it is a risk thing, right? Because if you're trying to do the clinical endpoint, we understand that's going to take time and effort and energy, but everyone knows how to do those. So again, I guess it does come down to a business decision, which way you'd like to go, but the agency considers every application as the data that it gets submitted to us. So we don't consider one more approvable than the other. It's all a matter of the evidence that you provide to us. And the more that you discuss with us, the more likelihood, you can have some success in whichever route you decide to take. Thanks.
Thank you. Another online, please.
Question for Dr. Zhensu. PSGs that recommend alternative study in lieu of the CCEP study, it seems that the RA-PSD has been recommended in all ANDAs, but other studies such as dissolution, particle morphology, charcoal block PKBE studies are selective and do not appear in all PSGs. Why is that?
Can you turn the mic on, please?
Sorry.
Thank you.
Yeah, thank you for the question. The realistic APSD is actually an advanced form of the conventional APSD that described in the USP compendium. So it's a more sensitive and reproducible and cost-effective method because it's an in vitro test than the CCEP study. And the advantage of this is it provides a more clinical relevant condition compared to the compendium APSD. And then this highlights the APSD, the realistic APSD is a critical study in the alternative study approach. So one of the important features that realistic APSD can avoid the actuation breathing coordination issue when it is tested with the actuation and the breathing has that synchronization. So So the realistic APSD, of course, it has its advantage, but also has its limitations. One of the things that we can foresee is the realistic APSD that when we test for example, the low specific resistance DPIs, it may exceed the flow rate of 100 liter per minute, which exceed the inertial sampling apparatus, the validation limit. So yeah, because the required peak respiratory flow rate, for a cascade impaction operation, and that's one of the reasons that the test is included in as one of the weight of evidence approach, but it's an important one. Based on like other alternative study, like the dissolution method, for example, it's also a pivotal study recommended in the PSG. But for those like the highly soluble and rapid dissolution profile drug, it may not be feasible for the dissolution testing. Yeah, thank you for the question.
Thank you. Dana, yeah.
Yeah, can I just add that? So aerosol performance for any inhalation product is applicable. So I think that's why you see realistic APSD testing in almost all the option 1 based approaches. It's applicable to almost everything because aerosol performance plays such a critical role, and clinical relevance is always key for us. But when it comes to other approaches, where we try to stick dissolution versus permeability, we really want to understand what's going on at the bioavailability of the site of action. If dissolution, your drug's slowly dissolving, it's going to be moving around, then the dissolution is probably the primary factor that's playing a role in that bioavailability. And that's not the case for every product. Sometimes you have something that's fastly dissolving, and your permeability is going to be the thing that's dictating its absorption properties. So that in those cases, we don't necessarily think that the dissolution test is going to tell us anything that's as relevant. So that's where those morphology characterizations fall in that place too. Sometimes morphology plays a critical role, how it becomes bioavailable. Sometimes in morphology that doesn't play as a big of a role. So I think that's where you can see the guidance getting more product specific, because really we are trying to understand what's going to be the most critical product performance parameters that are going to affect that bioavailability. Thanks.
Thank you very much. And I think we've run out the clock on questions. So thank you very much for our panel.